But first of all, I would like to thank the Pontifical Academy of Sciences and the Chancellor, and of course, Professor Coppens, for the very kind invitation of coming here and the very excited and stimulating conversations we had with this outstanding company of colleagues. And of course, the very, very stimulating, extraordinary environments that we're in. I'm going to present ideas from a collaboration with Jean-Pierre Changeux that originated from a chance encounter at one of the meetings of the Human Brain Project. Some of the ideas of Jean-Pierre Changeux were already mentioned during this meeting, for instance, yesterday. So I think he's going to be here in spirit. And our collaboration also involved a postdoctoral fellow from our institute in Hamburg, Alexandros Goulas. And there's already a published paper of which I will present some key ideas. It's a very long paper, so I'll just excerpt some of the features. And I should put the disclaimer, I'm not going to talk about where religion comes from. That would be preposterous, I don't know. I'm going to talk about what we understand in terms of the organization of the human brain, in particular the connectivity organization, and what implications for the computational aspects of that connectivity and for the function of the brain that may have. And so I'm guided by this key statement of Aristotle, who's been called the first biologist and the first philosopher. He collected a couple of firsts, and he stated the unity of structure and function by saying, the thinkers, however, to whom we are referring, attempt to state the nature of the soul only. With regard to the nature of the body, which is to receive the soul, they determine nothing in particular. And thus, although every body seems to possess a distinctive form and character, they act as if it were possible for any soul to clothe itself in any body. So I'm going to talk about the peculiarities of the body, and then I'm leaving it to you also to draw inference on the nature of the soul that may be clothed in that kind of body. But what aspect of the body should we be talking about? There's an apparent paradox of the very rapid expansion of key human cognitive abilities, including a very large and stable long-term memory, as well as a very large working memory capacity, theory of mind, cognitive thinking, which is very flexible, language, which we heard about, appreciation of beauty, culture, and spirituality over a relatively short time span, something that appeared in fewer than 2 million years. This expansion proceeded against the background of apparently only few changes in the number as well as the kind of genes found in the human brain when compared to other mammalian and primate brains. So here we see that we don't really have more genes than the mouse, and not really so many more genes than the fly. And also, if you look at the nature of brain protein coding genes that are involved in neural substructures as well as in coding for synaptic organization, they are found more conserved for brain structures than they were for other parts of the body. So this is the totality of all genes. They're more conserved over here. This is for the brain. And in fact, the most variable genes are found for the testes. Single cell sequencing had put more variation on the scheme, but in particular the cerebral cortex, which we often associate with cognitive thinking, is actually found to be very conserved. Cerebellum is actually more varied, and there may be more specifics in the human cerebellum than in mouse cerebellum, for instance. So we have largely conserved primate genes, and there may still be unidentified discrete gene changes from Australopithecus to Homo sapiens. We don't know about that, but so far there hasn't been a smoking gun, a single gene that directly explains the human capacity for advanced cognitive abilities. However, something that is much more easy to explain is there may be plausible changes in gene expression rather than in the appearance of totally new genes. So we take what we have and modify it a little bit in modifying some of the existing properties. And so we have a parsimony of a genetic envelope of the primate brain that is extended for the human brain. And so our connectomic hypothesis attempts to link gene evolution and connectomic evolution and explain its impact on human cognitive abilities, because the connectivity of the brain in recent years has emerged as a very helpful intermediate functional phenotype that sits between the genetic level and is being defined by networks of genes, not individual genes. And also, as a network, helps to explain cognitive functions in the combined emergence of cognitive abilities. And we make a distinction between features that are scaled in the primate envelope versus potentially singular features. So I'll put here the relationship between brain mass and the number of neurons, where Hakolana Hauser, for instance, has suggested that we don't have an exceptional brain, that some of the relationships are just scaled up. That doesn't mean that they don't result in peculiar consequences in our brains and abilities that are special, but they are readily explained within the primate envelope by just saying that we have larger brain size. And there may be some other features which are more specific to the human brain, which is not to say that they're not genes that already exist in some other primates that are just overexpressed in the human brain. So a simple modification of that kind would just be prolonged development. And we already touched on this subject, and Stanislas Dehaene in particular just mentioned the expanded cortices of the dorsolateral surface, so association cortices of the parietal cortex, the temporal cortex, and the prefrontal cortex. And it's exactly these cortices that are expanded in human development, and they're also expanded in the evolution if you compare macaques and human brains, and the correlation between them is very high. So a simple suggestion would be that we have a modification of genes that prolong human development. So for instance, in the comparison of the human development with the mouse development, the human development is much delayed. And because of the exponential growth of neuronal progenitor cells, just a few extra weeks of gestation compared to other great apes makes all of a difference in producing much greater neuron numbers. So we have perhaps 1 or 2 or 3 weeks more than gorillas or orangutans, 38 weeks as compared to 35 plus weeks in other monkeys. But that really makes all the difference that we have all of a sudden 100 billion neurons and no longer 6 billion neurons as you have in rhesus macaques. And with that dramatic increase in the number of neuronal elements of the human brain, which of course are potential representatives of long-term memory traces, we also have an increase in the number of areas, not quite as dramatic as for the number of neurons. Theoretical considerations suggest that the number of areas should scale with the square root rather than directly linearly with the number of neuronal elements. So you have in the latest parcellations about 180 cortical areas of the human brain as compared to perhaps 100 or so in the rhesus macaque. But the most dramatic change, of course, is in a very large increase in cortical connectivity. So not just in the number of neuronal elements and number of neural areas, but in fact, you have many more pathways, for instance, in the human prefrontal cortex than you have in the mouse due to this prolonged development. And much of what we have in the volume increase of the human brain is actually the white matter connectivity reaching between different cortical areas. So there's a dramatic increase in absolute connectivity, which of course has consequences for the combinatorial abilities of the human brain. But what I think is more relevant even is that the increase in absolute connectivity goes along with the sparsification of the connectivity. And the reason for that is that as you increase the number of neural elements, you cannot keep them completely connected due to volume constraints. So if you were to connect all the 100 billion neurons in the human brain, you would end up with a brain the size of a planet. And so rather than maintaining proportional connectivity, which is impossible due to these constraints, we have near constant connectivity. So in C. elegans nematode worm, you have perhaps on average 10 nososynaptic contacts of the 302 neurons. In a human brain, you may have between 1,000 and 10,000. Now that's more, but it's not more as compared on the scale of the neural elements that you have. So there's a relative sparsification of the human brain due to these volume constraints. At the same time, we have a reduction of the relative projection length. So we have an absolute increase in projection length, but if you put projection length relative to the size of the human brain, the connections actually get shorter. There are beautiful studies, cross-species studies from the group of Henry Kennedy that demonstrate that very clearly. And they may have to do, or they may arise from spatial temporal growth patterns where you just have a more local window of opportunity of connecting randomly with your neighbors, and due to the larger area of the brain, that's more restrictive on a local scale. So what we find in an expanded brain, and that would be general relationships for any increased brains, is that you have a near constant node connectivity resulting in network sparsity, and that you have reduced relative projection length, which enforces more local connectivity, and both of them combined produce an increased modularity of the brain. So we have a number of elements that are increased. So you have an increased storage capacity. If we think about language, that may result in an increased vocabulary. There are findings that cognitive abilities, as some general intelligence measures, actually scale with the number of neurons across different species. And we also have increased modularity. And this modularity erects some barriers between the putative modules. And it acts also in order to stabilize the different items that we store in the vocabulary or in the memory in general. And so theoretical studies have demonstrated that increased modularity is actually helpful for enhancing self-sustained activation, information transfer, and working memory capacity. And here's a computational study by Rodriguez and Waiwayan and colleagues that shows that as you move from regular networks to highly modular networks in an echo state reservoir computational setting, you increase the duration of the memory as well as the number of sequences that can be stored. So modularity can actually be quite beneficial to computational properties, depending on the kind of parameters that you use for the neurons. At the same time as the brains increase and you have modules, the argument of the number of neural elements that cannot be completely connected repeats at the next higher level. So what you have are in fact modules of modules of modules at various scales. So you have hierarchical modular connectivity, as shown here for the example of functional connectivity by Park and Friston, where you connect large-scale patches across different parts of the cerebral cortex. And within each of these, you have networks that are locally connected, which then consist of voxel-level networks, which are again connected. And this, we suggest, I mean, this is a naturally evolving structure. The larger the brain, the more pronounced this hierarchical modularity will be. And what we would suggest is that this kind of hierarchical modularity is a natural substrate for the kind of recursive processing that we heard about already this morning. Now, the details are not quite clear, but theoretical studies, such as the one by Moretti and Muniz, suggest that this is an architecture that naturally supports criticality. Criticality is a feature where the dynamical system is poised between a fixed point where not much interesting things are happening, where the activity results back to zero, and a chaotic behavior of the system. And as biologists, this is nothing special to us because the brain is alive and is at that boundary between chaos and no activity all the time. And so the physicists had some explaining to do. And one of the things they found that a hierarchical modular network architecture, particularly when sparse, supports that kind of scaling of activity. So there's the small items here, which are represented with a high likelihood. There are large items here. This could be activity. This could be duration between individual stimulus presentations. They're reported with a low frequency. And the fact that there's no particular scale, that this is a power graph and a scale-free organization, demonstrates that this may be a structure where you can combine items of different scales very naturally. So this may be a substrate for that kind of recursive language structure, or the recursive combination of symbols for language abilities such as merge or dendrophilia. There's another simple consequence of the expansion of the dorsolateral surface. So in terms of the networks that are projecting across the surface, the number of levels that are being used, the number of layers, is actually increased in the human brain as compared to other primates. And the simple consequence of that can be found in modern deep learning networks. And the simple invention, relatively straightforward invention that was made there, was just to add more layers to these networks. And that resulted in a dramatically increased performance at the higher levels. So shown here for a synthetic agent that has a much lower error at the higher levels. And it also results in more generalization capability. So I'm also referring to a model by Tomasello and colleagues, where they showed that at the intermediate levels, at a higher level, so to speak, there was much more general semantic representations than we had at the ends, at the sensory motor ends of this model, which was using a Hebbian learning structure in order to identify semantic representations, and there were category-specific representations at the sensory ends and much more general representations at the center. So expanded cortical gradients may result in increased depth of multi-level representations, which is reflected in improved accuracy and increased abstraction. So all of these things are still directly resulting from the size scaling of the human brain. So these are network consequences that simply result from increasing the size of the human brain. Now comes a more specific aspect, and this has to do with the cytoarchitecture. So it's not just that we get more cortical areas in the human brain, but we also get areas that are more cytoarchitectonically differentiated. So adding more neurons also means that the later developing areas are more differentiated in terms of the laminar structure. They also contain more neurons. And so one of the consequences of that is that for the global neuronal workspace, which I hope Stan Lehane may talk about, so I don't have to introduce the concept, but I think you're all familiar with it in any case, shows a differentiation between the core structure and the periphery structure. So we find a core-periphery network organization as a potential neural substrate of such a global neuronal workspace across many mammalian species, even including the mouse. But what is peculiar of the human brain is that the core and the periphery are cytoarchitectonically differentiated. So we have a different local cellular organization of peripheral areas and of central areas, core areas. And that may result in a specialization and an increased depth and specificity of representations that may also result in multi-scale abstraction. Now comes a very specific feature which we reported a number of years ago, which was rediscovered from somewhat obscure anatomical work in Germany in the 1960s by Friderichs. And that goes under the term of external pyramidization. So German anatomists love arcane words. And it's the observation that in the laminar organization of cytoarchitecture of the cerebral cortex and of laminar projection patterns, there's a very distinct shift of what's going on. So another characteristic feature of the cerebral cortex is that laminar organization. And there's a varying degree of laminar cytoarchitectonic differentiation. And tied to this differentiation is a relative location of projection neurons with areas of high differentiation. So there's some gradients shown here which go from low differentiation to high differentiation, actually showing superficial cortical origins of projection neurons. And that has been termed external pyramidization. And there's some consequences that the larger pyramidal neurons are located in the superficial layers rather than the deep layers of these areas, because larger projection neurons also have longer axons, and they have increased Booton size. And so they may be thought of as the highways of information transfer in the cortex. So they're really very powerful projection neurons with a very powerful drive. And it's important to know where they're located. And what we see when we compare across different mammalian species here, for instance, for the rat and the human brain, then we find a shift in the human brain to more areas being highly cytoarchitectonically differentiated and having the larger projection neurons located in the superficial layers. So for instance, anterior cingulate in both cortices is relatively undifferentiated, but the lateral prefrontal cortex in the rat and the rodent is still relatively undifferentiated, but it's highly differentiated or more differentiated in the human brain. And so the projection neurons have moved to the superficial layers. So that external pyramidization happens to well-differentiated dense areas. And in the lab internal slang, we call that the rise of the pyramids, because the pyramidal cells move to the upper cortical layers. And so you can see the shift here, particularly when compared between the rhesus macaque monkey and the human brain. So that may have some consequences to the ignition of the global neural workspace, for instance, because it's mostly the areas in the periphery that project back into the core that have become more powerful in the human brain. And it may also be important for the supragranular control of working memory, because it's known that working memory in terms of the frequency bands, it's mainly sustained by deep cortical layers, but it's the upper cortical layers that select the contents. There's some very nice reviews on that. And so the powerful control of the supragranular layers may help to identify items in the working memory. It's interesting to note that it's also the supragranular layers, the upper cortical layers, that have a particularly delayed development. So here, compared from the human prefrontal cortex, are neurons from layer 3C, upper cortical layers, with layer 5. And there's a delayed growth spurt of the upper cortical layers to a period of 2 to 5 years when they mature fully. And that, of course, is a very interesting and critical period where language recursivity and theory of mind and many other crucial cognitive features of the human brain develop. And they can be frequently associated with the upper cortical development, not so much with the deeper cortical development. And we already touched on verbal expression of this kind of diagram yesterday, that generally the human brain shows a much prolonged postnatal maturation, which of course opens up a window of exposure to cultural influences, to teaching, to very close contact with other social beings, and of course to the transmission of religion ultimately. So there was a lot of information, and this looks confusing, but in fact I present this in order to make it perhaps a bit more palatable and a bit more unified, because it turns out that several of the features that I was talking about are actually aligned across the cortical spectrum. So what I show here on the left is with the same representations from a very nice recent review by Sidnor and Tom Satterthwaite's lab, many different features of a progression of cytoarchitectonic gradients, evolutionary expansion, metabolic gradients, gene expression, and so on and so on. And it seems that they are not perfectly, but roughly aligned across the surface. So it's the same kind of cortices that we were talking about, that are late developing association cortices of the dorsolateral surface, parietal, temporal, and prefrontal cortex, that share a number of features. And of course, this combination of features, or this unification of features, may be achieved by developmental mechanisms that at present are not fully clear. We contributed to this unification by showing that also neurotransmitter receptors, based on the work of the late Zillis in Jülich, are associated with these gradients. So for instance, there's an increased diversity of different neurotransmitter receptors along this axis. There's an increase in the ratio of excitable to inhibitory populations across the same axis. And also there's a ratio of metabotropic, modulatory versus ionotropic, more fast-acting receptors along the very same gradient. And on the right-hand side, I have an image from the non-human primate brain, which is organized along the gradients of cytoarchitectonic differentiation, with the high differentiation on the outside to the low differentiation, lower number of cells on the inside, which shows that this also aligns with connectivity. So the more green you have, the more the connectivity is aligned with the similarity in the cytoarchitecture that's being connected by these projections. And the red dots here are the ridge club, or the network core of these areas, which is associated with the low cytoarchitectonic differentiation in a smaller number of cells. So there's a broad alignment of structural, connection, and functional features along a common cortical spectrum, from outside areas, high differentiation, to inside areas of a low differentiation, and a number of other features. I don't really want to go through this table in great detail. So typically people think of an axis of cortical organization stretching from sensory areas on the outside to association areas on the inside. But I think perhaps a helpful conceptual simplification of that already simple idea would be to distinguish between outside areas and inside areas, which have a number of different properties in terms of their connectivity, in terms of their neurotransmitter receptors, and importantly, in terms of the maturation. So it's the outside areas with their high spatial temporal resolution, because they're on the interface with the external world, and that have a more limited plasticity, and they mature more early in postnatal development, as compared to the core areas that are more flexible in their intrinsic organization, that also have more neuropil and have prolonged plasticity, in particular in the supragranular areas. So to put it all together, here is how you can conceptualize the expanded cortical spectrum of the human brain in terms of just adding more rings of outside areas, because it's the outside areas that develop latest in the human development. And that results in a number of important features. For instance, the expansion of the outside areas gives us a much larger sensory motor interface, which helps us to represent the world, but also helps us to interact with the world in more fine-grained interactions. And in particular for language, and in particular for writing, that's really an essential foundation. There's also segregation of the outside areas so that they no longer communicate directly in the human brain, but they connect via or communicate via the network core. So that enforces a conceptualization of the sensory representations. And of course, we have a much expanded network core, which is now cytoarchitectonically differentiated and which has a much prolonged maturation in postnatal life. So that can lead to the acceptance or to the adaptation to cultural influences in particular. Okay, so there's a grand finale, so to speak, of what I tried to present you with. And we tried to go in this working hypothesis paper. It's really just a working hypothesis at this point, from certain biological premises, which may either operate within a primate genetic envelope or may represent potential singular features of the human brain, resulting in connectomic fundamentals, which then have functional implications of the various kinds I outlined. And in particular, the basis can be achieved very simply by modifying or tweaking just a few switches of gene expression. So there are many genes, for instance, that are being downregulated in primate development, and would just be enough to not downregulate it and to prolong the development just a little bit further. And then you would have this expanded brain size and differentiation given as a result of that. So at the moment, it's not quite clear what genes you have to modify. There's some candidate genes. There's some exciting work with transgenic animals, for instance, where people have expressed them in the macaque brain and actually found that this leads to delayed maturation of the supragranular layers and even increased working memory in these transgenic animals. So there's some strong clues as to what may have happened. But the point I want to stress here is that this expanded size of the brain, which I really thought was the foundation of everything that followed, is in particular important for changes of brain connectivity, such as the increased connectivity and thus enforced sparsity and hierarchical modularity, the increased representational depth of the networks that we possess, and an increased architectonic segregation of the core-periphery structure that's underlying the long-range connectivity of the global neural network space. And I would very boldly suggest, and that of course needs to be tested in much further work and also in computational work, that these features result in increased lexicon and learning and stable representations and potentially the scaling of activity as it's required for recursive processing, abstraction, control of working memory, and so on. But we can't stress enough the very important role of synaptic epigenesis and the very prolonged postnatal maturation, which offers us the chance for interacting extensively over many, many years and over thousands and 10,000s of instances with other human beings, with other cultural predecessors and artifacts, which may underlie language acquisition as well as acquisition of culture and spirituality, and which ultimately are also important, of course, for the diversity of individual brains, because we're all individuals due to the individual trajectories that we're being exposed to. Thank you very much for your attention.